Hello, I'm Dan Maltbie. I'm the CTO of Annai Systems, and it's a small company here in Silicon Valley. We focus on genomic data, how to move it, store it, secure it, compute it, find it. And what I'm going to talk about today is a project that we've been working on in conjunction with UCSC and the National Cancer Institute. My collaborator, Chris Wilkes, is here today, and we have a poster outside. We can go into details. But I want to describe a little bit about the system, and then I'll talk about what I think is really one of the fundamental problems in terms of moving forward. If I can figure out how to move this. So this is what we've been focusing on is a project for housing the largest collection of cancer genome sequences for the Cancer Genome Atlas project, which is a project funded by NCI. UCSC is the operator and manages the system. Annai Systems provided the technology and implementation of the system. And it currently has over 300 terabytes of sequence data, over 24,000 sequence files in this system. And the interesting thing about this project is it involved really a collaboration across a large number of organizations. So this was really a major change in the way research is done and the way research around cancer, in that it became a collaboration among multiple labs, multiple researchers, rather than one lab doing everything from the beginning to end. And what I mean by that is that, for instance, on the top left there, you see something called genome sequencing centers. There were sequencing centers that were identified that their job was to do production of genome sequencing. These are world-class sequencing centers like Broad, Washington University, Baylor, MD Anderson, and these folks were potentially, essentially contracted by NCI to produce sequences. So their job was to ensure that what tissue sample was received produced a high quality raw and mapped sequence that could be used downstream by analysis centers. That data is collected at the CG Hub, which is located in San Diego. This is the location where we have a high capacity. It's designed to be a multi-petabyte system, high performance I/O. Because these are large files, we designed the system to be able to move the data very, very efficiently in and out of the system. And again, if you want more details on the system, we'll talk about that outside. Once the data is in the CG Hub, there is a whole host of researchers that access it. There are specific analysis centers that do primary analysis on this data. And there's over or almost 300 researchers that use this data either for NCI-funded projects or other projects outside of that. So this created a lot of interesting characteristics around how to be able to produce and reuse data. So this sequencing information is used not only for a primary research project, but is reused and repurposed by other researchers for other projects. And so that means the data has to be in a form that's reusable, that's understandable, that can be repurposed, which I think is another unique characteristic around this system. And so that sort of leads me to where things are going. Whoops. So I think this is the evolution of what we'll see in genomics and medical systems, is that this idea that there will be a lot of different repositories. Someone mentioned the 1,000 genome system, the Washington U St. Jude data set that was produced recently with 600 cases of pediatric cancer, which is being used by other researchers to make discoveries. So there'll be a large number of genome collections that will be out there. Some of these will be public and widely available, like 1,000 genomes. Some of these will be protected, like the TCGA data set. If you're an authorized researcher, you can have access to it. But since it does represent consented data, it has to be protected. There may also be private data sets, hospitals, clinics, other locations that would produce and collect their own sequencing data and maintain that. Ultimately, there may be commercial data sets where you can go get interesting, unique genome sequences that would be used for studies. This set of data is then going to be used by a wide variety of analysis systems. I think what we're going to see is really an explosion in the analysis space, because there's going to be lots and lots of people who want to do different kinds of analysis. So this creates really an ecosystem, a collaborative ecosystem that people want to use a lot of different things. And so what it leads to is what I think is the fundamental problem, is that we lack good standards, we lack good ways to share this information. this is because it's an early industry, it's just growing, but we think this is where it has to go. So Ethernet had IEEE, IETF has the internet. Genomics needs genomic phenotype expert group. And I'm done. And I'm thrown off the stage.